The heart of NeuroDOT is the processing pipeline. These operations are implemented as a collection of toolbox functions which transform raw optical data into a volumetric reconstruction of brain activity ready for visualization and statistical analysis. However, it can be challenging to understand the pipeline workflow, especially for a new user. This video provides a brief overview to help you get oriented. We introduce the standard NeuroDOT pipeline and explain the organization as it relates to the objectives of data processing. The goal is to show how the individual processing steps and the individual tutorials fit into the big picture. This will prepare you to work with the NeuroDOT instructional materials that cover these topics in detail. To begin, consider a typical optical imaging study. Here we'll assume a task-based experiment, but the workflow we will describe applies equally to the processing of resting state data. The goal is to construct a map of the neural activity that occurred during data collection. In optical imaging, the primary data comprise measurements of light intensity provided by optical detectors on the scalp. A collection of emitter detector pairs is sampled over the duration of the experiment. The number of available signal pairs is determined by the optode array configuration. But because signal quality degrades with increasing separation distance, not all emitter detector pairs will provide usable data. This is revealed by a data quality assessment that is performed at the beginning of processing. Once QA is complete, the data enter the processing pipeline. In the broadest sense, processing consists of two transformations. First, the pairs of emitter detector time series data is converted into a spatial map of optical changes that occurred in the brain during the experiment. This reconstruction represents the bulk of processing, involving a series of filtering and regression operations, as well as construction of a computational model of light propagation inside the skull. A second transformation converts this optical map into concomitant changes in neural activity. Spectroscopy has established that oxygenated and deoxygenated hemoglobin differ in their wavelength-dependent optical properties. Thus, active brain areas exhibit a change in optical properties due to recruitment of oxygenated blood. This effect can be exploited to infer changes in local neural activity, given changes in local optical properties. Having described this processing in general terms, we now present the NeuroDOT implementation in more detail. This will allow us to identify video and PowerPoint tutorials that are relevant to specific processing tasks that appear in the pipeline. In this figure, data collection is shown at upper left and feeds two workflows that converge to produce a tomographic map of brain function shown at lower right. The first of these workflows applies a standard series of pre-processing operations to the data. Some of these steps are self-explanatory, such as bandpass filtering, whereas the motivation of other steps may be less apparent. Here we only state that these operations reflect best practices established during the development and application of NeuroDOT. The second workflow entails construction of a computational model of light propagation inside the skull. This is based on a three-dimensional finite element mesh, taking into account the geometry and optical properties of the brain and surrounding tissues. As part of this analysis, the position of the optode array must be fixed relative to the finite element mesh. This is called array alignment in the documentation. A standalone app is provided in the distribution that is used to perform this operation. The end result of light modeling is a matrix which is applied to the pre-processed data to obtain a reconstruction of optical changes in the brain. Spectroscopy is applied to convert this to a map of brain activation, which is then typically normalized to a standard space to permit group analysis. The reconstruction can now be reviewed using NeuroDOT visualization tools and is ready for statistical analysis. This can be performed within NeuroDOT, or by saving the results as a 4D Nifty file and using an external tool such as SPM or FSL. The NeuroDOT tutorials are organized around the operations that appear in this flowchart. The data processing tutorial provides a detailed review of the core NeuroDOT workflow, including pre-processing of array data, use of a light model to obtain a regularized reconstruction, and application of spectroscopy and spatial normalization. The data quality assessment is presented in a companion tutorial. Videos specific to light modeling include coverage of the NeuroDOT array alignment tool and a conceptual introduction to computational light modeling explaining how and why finite element analysis generates the model used in reconstruction. Finally, the visualization tutorial covers the tools available for viewing results, including both sectional and surface renderings of volumetric reconstructions, as well as parametric maps. Additional videos are available that cover background topics, such as installation and setup of NeuroDOT, installation of the near-faster light modeling package, and loading data, including data from websites such as OpenNeuro. Hands-on exercises are provided by the PowerPoint tutorials included as part of the NeuroDOT documentation. Their organization is similar to the video tutorials, although some additional or alternate selections which cover specialized processing tasks are provided. See the tutorials directory in the NeuroDOT distribution for a complete listing. This concludes our overview of the NeuroDOT tutorial series. If you are new to NeuroDOT or to optical imaging, you may want to view the entire collection in the order shown here. This will provide an accelerated introduction to the subject and to the software, from installation to visualization. Alternatively, if you are simply looking for information on a specific topic, individual videos may be reviewed as needed. They are intended to complement the PowerPoint documentation included with NeuroDOT, offering background and context for the associated hands-on exercises. Additional information may be found in the resources listed here. Feel free to contact the development team with questions or post them in the comments section below. Thank you for your interest in NeuroDOT.